Hello, I'm Jill O'Donnell Tormey, CEO and Director of Scientific Affairs at the Cancer Research Institute. I'm very happy to be here today. And we have at CRI partnered with Focus Ultrasound since 2015, and we're continuing our partnership, and I look forward to the meeting. So I've been asked to just give a little background about CRI for those that may not know who we are. Our mission is to save more lives by fueling the discovery and development of powerful immunotherapies for all types of cancer. This is a mission that we have held on to since 1953. This is way before the wave of excitement that immunotherapy has taken place over the last, 10 to 15 years. We are very proud of the foundational work CRI has done at a time when no other organization was really supporting immunotherapy. We funded more than 3,500 scientists over this time and actually invested almost $475 million. And I think it's thanks very much to what CRI has done that we are where we are today in immunotherapy. We have four mission pillars. Really, one is to power basic and translational research, which is where all the discoveries come. And this is where all of the future therapies come from. It always goes back to basic research. We also try to accelerate these discoveries into the clinic with an innovative collaborative model, which I'll speak a little bit further on in the next slide. We also play a role of convening thought leaders and bringing together really the key people in immunotherapy to discuss major topics and really guide the field. And last but not least, we have become a major source of trusted information for patients and caregivers that are really interested in understanding in immunotherapy and what role immunotherapy may play in their potential treatment plan. We have a full spectrum of research programs. Our oldest program is our Irvington Postdoctoral Fellowship Program, where we really support young scientists, both PhDs and MDs, that are working in the laboratories of senior scientists. This is where we really support most of the basic discovery. We also have a translational research program, the Clinical and Laboratory Integration Program, which is this bridge between the laboratory to the clinic. And this is where we hope discoveries are made, either taking patient samples back into the clinic or developing models in the lab that can mimic the clinic so that we can really inform the next generation of immunotherapies. Our Anna Maria Kellen Clinical Accelerator is a unique nonprofit, academic, industry collaborative model that serves as an incubator for multi-center clinical trials that are testing a variety of cancer immunotherapy combinations. It relies on a global network of about 100 clinical investigators that really inform us of important questions that they feel need to be asked in the clinic and are not being yet asked. CRI then prioritizes those ideas and those potential protocols and really works to go out and seek not only the drugs that need to be combined that are owned by biotech or pharma, as well as we bring together clinical trials management with our other non-for-profit partners, and we provide financial resources so that these clinical trials can take place. As I said, they really are answering scientific questions, so I think it's really to de-risk ideas and hopefully inform and bring these in to the development pipelines of major pharmaceutical companies if the data that comes out of these trials proves important. And it is fueled by a venture philanthropy fund, so it's a way to hopefully get returns on our investment and keep the program going in an evergreen way. More recently, we have developed a Technology Impact Award, which actually, unlike all of our other programs, we're really focused on biology, are focused on technologies in immunology that could hopefully develop these new technologies that could be impactful for and adopted by the entire field. And then last but not least, our Lloyd J. Old Star program, which is Scientists Taking Risks, is focused more on like a Howard Hughes type model for mid-career scientists, which we define as late assistant professors to early associate professors. And it's long-term funding. It's a 5-year, $1.25 million grant that is not tied to a project. So these stars, or the people we believe are going to be the future of the field, can use these dollars to do high risk, high reward, new questions to be asked that maybe they don't have funding from their R01 grants. I think one of the greatest assets CRI has is our Scientific Advisory Council. It is a large group of 77 members that includes 4 Nobel laureates, one being James Allison, who is our 2018 Nobel laureate, as well as 27 members of the National Academy of Science. These are our talent scouts that have done a great job of really selecting most outstanding scientists and projects to support, and are really, I think, responsible for the impactful role that CRI has had over the years on the field of immunotherapy. Just a little bit beyond CRI, for an audience that may not be familiar, I just wanted to reiterate that there are many types of immunotherapy. I think when people talk about immunotherapy these days, they only think of checkpoint inhibitors, but that's only one type of immunotherapy. We look at CRI as immunotherapy into 5 buckets. Adoptive cell therapy, which includes the chimeric antigen receptors and other cellular, like tumor-infiltrating lymphocytes. Cancer vaccines, which are both preventative and therapeutic, various immunomodulators, including checkpoint inhibitors, oncolytic viruses, and also targeted antibodies, many of them like the monoclonals that have been around for many years, like rituximab and such. So these are all areas, and to be quite honest, the FDA has really only improved the chimeric antigen receptors, as well as checkpoint inhibitors, being the anti-CTLA4 and a number of anti-PD-1s and PD-L1s. They've also improved TVEC, which is an oncolytic virus for melanoma, and of course, these very monoclonals that are given as monotherapies for many, many years. But all these are areas of active research, and I think we still need to come to learn how these all work together along with chemotherapy and radiation, and perhaps a focused ultrasound in terms of developing a really impactful immune response that can deliver effective immunotherapies for all types of cancer. There are some key areas I think the field is focused on right now. Understanding how tumor cells evade or suppress the immune response is an important area of research, as well as finding solutions to checkpoint resistance. We've come to learn that as impactful and as effective as checkpoint therapy can be in a variety of cancers, it doesn't work in all cases. In the best case, 50 to 60% of patients, maybe in melanoma, respond, and in other cancer types, maybe only 20%. And we need to understand why there is an innate resistance to checkpoints in certain tumor types and in certain patients, and well as adaptive immune resistance in terms of patients that start to respond but then start failing. And so that's an important area because, as I said, we have the proof of principle that your immune system can definitely be used against cancer, but if this resistance exists, it's one of the things we have to overcome. Also important is predicting responses to IO. This is biomarker research. In terms of understanding, can we develop and understand biomarkers that at the time of diagnosis, a patient could have these biomarkers and say, this is the exact right combination of drugs that you need to give to this patient to get an effective immune response that will put their cancer in check. And lastly, all of this together really is helping us to design rational immune-based combinations, which I do believe is the future. And so talking about the future, I think it can be put into 3 buckets. There are mechanistic discoveries that are happening in the laboratory on the immune system itself, but also in genomics, on the stroma and the tumor microenvironment, which is where the action is and where the immune system has to ultimately work to eliminate cancer. The role of metabolomics and the microbiome, all of these are affecting the immune response and the immune response to certain treatments. So we need to understand that. I think correlative science, which I describe as really doing that deep dive in patient samples being treated with either standard of care immunotherapy combinations or in clinical trials. And this is where we will develop the biomarkers that will really inform patient selection. We can work on optimized combinations that include checkpoints. We're going to go beyond just effector molecules that affect T cells. And this all requires a lot of bioinformatics because with all these deep dives, analysis and assays that we do, you develop an awful lot of information that needs to be really analyzed in a way that can really inform what is the next step to take. And ultimately, putting all of this together, we'll get to personalized IO combinations, which I do believe is the future. Another future is using gene editing and synthetic biology to make better cellular therapies that hopefully can work in solid tumors and not only in hematological malignancies. And then, of course, there's additional work on developing effective therapeutic vaccines. So a lot to do, but I think the future is bright. And if we continue to support research and excellent science, we're going to get there and get better immunotherapies for all patients. So thank you very much. It's an honor to be here, and I look forward to the discussions the next 2 days.